welcome everyone. I'm Matt Mosconi. I'm the director of the Kansas Center for Autism Research and Training, and welcome to the 2022 Autism Across the Lifespan Conference. My goodness, we're in person. Jeez, yeah, clap. Go ahead, we can clap for that, all right? It has been 4 years since we've done this in person. We've done a couple of virtual online meetings, and I'm just so glad to be able to reconnect here today, live, making social contact. And if I see you, I haven't seen you in a while. A couple people already said hi to me. I recognize most people, but sometimes I just see eyes. And so please, let's reconnect. It's great to see everyone here today. And thank you so much for braving the weather and joining us today. We have a wonderful program planned. I'm going to get off the stage in just a few moments. My primary task is to introduce our wonderful keynote speaker, which I'm very excited about. I'm hoping you are both excited about it as well. But I do have a few notes before we get started and before we get moving into our actual program. The first is we strongly encourage people to wear masks today. I've taken my mask off just for speaking purposes, but I'll be wearing it the rest of the day. I know people have differing opinions on these things, but we're just trying to stay as safe as possible, particularly among people who may be more vulnerable related to the pandemic and to COVID-19. So please be respectful of each other. We strongly encourage masks. If people don't have masks with them, that's okay. We have masks that should be circulating at some point around the room. If not, they're in front as well. But again, I ask everyone to please be respectful of each other's wishes around this, and we'll be as safe as possible today. I'm also asked to provide to highlight the fact that we have surveys at your table. 2 points on that front. One is we look at every one of these surveys, I promise you, and they shape our meetings, our future meetings. We read each comment, we look at the feedback, and it drives what we do and how we program this conference moving forward. There's also a question on there related to the world we live in today, and that is one where COVID-19 exists. And it asks about whether we want to do hybrid, whether we want to do in person, whether we want to do remote, or all of the above in the future. And we're trying to figure this out as we go. And we're trying to do this in communication with each of you to do what is best and to do what is most helpful to the community. So please give us your feedback. Let us know what you think, and we will be reading it. Before I introduce our keynote, one more note. I want to do some thank yous, because the reality is that this meeting is a lot of work. It's a lot of fun to happen. Some of the work is fun in terms of preparing for this meeting, but a lot goes into it. And I wanted to express my gratitude to the speakers for today, who have generously devoted their time to not only doing the presentations, but preparing this and sharing some of their latest work for the meeting. I also wanted to thank Farmer's House for doing the catering this year. They've done a superb job for us in the past, and they've gone the extra mile, and we're doing brown bag lunches today, again, in the interest of safety. But I wanted to thank them for supporting this meeting. Dustin Boehm and the Edwards campus for hosting this meeting and providing us with this venue to get together. And then also our planning committee, including Renee Jameson, Drew Rossdale, Hillary Casey, and Jen Humphrey, who were each involved in preparing this meeting, are involved today in recording and doing the IT work on this meeting. And then the leadership of that committee, including Catherine Unruh, who has worked tirelessly to help structure things. And many of you know Sean Swindler, who always forgoes sleep for about a month to make this meeting happen and to make sure that it is smooth and successful. And so I did want to give each of them a round of applause. And now I'll get to it. So it is my pleasure to introduce our keynote for the 2022 meeting. Jeremy, I think he can hear us. I know we can see him and we can see his slide. Dr. Jeremy Veenstra-VanderWeele will be speaking with today from the other Manhattan. Many of you already know Jeremy's work. He's a leader in advancing new therapeutics for supporting autistic individuals. He has more accolades than I have time to describe, but I'll be succinct and talk about some of his accomplishments. Dr. Veenstra-VanderWeele is a Suzanne Crosby Murphy Professor of Developmental Neuropsychiatry at Columbia Irving Medical Center in New York, Director of the Division of Child and Adolescent Psychiatry at New York-Presbyterian Morgan Stanley Children's Hospital, New York State Psychiatric Institute, and Columbia University, and co-director of both the National Institute of Mental Health postdoctoral fellowship program for translational research in child psychiatry and the Whitaker Scholar Program in Developmental Neuropsychiatry at New York State Psychiatric Institute and Columbia University Medical Center. And now you know why I have notes for this keynote. Dr. Veenstra-VanderWeele is a child and adolescent psychiatrist who uses multiple research tools in pursuing new treatments for autism spectrum disorder and pediatric obsessive compulsive disorder. And he's the rare scientist. I don't know many people that do this, and I don't know anybody who does it as well as Jeremy. But he leads studies of both genetic mouse models to better understand causes and new therapeutic strategies for autism. But then he actually leads the clinical studies evaluating these therapeutics with patients, with individuals doing human studies. Add to that the fact that he's practicing psychiatrist and center director. It's hard to imagine when he sleeps. Dr. Veenstra-VanderWeele conducted his research fellowship training at University of Chicago, working with Ed Cook, before doing postdoctoral training with Randy Blakely at Vanderbilt University. He was a faculty researcher at Vanderbilt for multiple years before moving to Columbia and taking on his current roles in 2014. His work is supported by numerous NIH, foundation, and industry awards. He was recognized with the Blanche Adelson Award for Research in Child and Adolescent Psychiatry from the American Psychiatric Association, a very prestigious award. And by any metric, it is clear his research is having a significant impact both on the scientific community and directly on individuals with autism. For those research scientific nerds out there in our field, his H-index is 60 and his I-10 index is 119. He's been cited over 12,000 times. For those who aren't research nerds who follow these statistics, that's pretty awesome. On top of all that, I can attest he's a pleasure to work with. He's a wonderful colleague, a wonderful collaborator, and his research is having a real impact, and it's one that many of us admire greatly. He's an all-star in our field, and it's my honor to introduce him as our keynote speaker this year, Dr. Jeremy Veenstra-VanderWeele.
Thank you so much, Matt. It's good to be with you all. I wish I could be there in person. I will say, in particular, that was such a generous introduction. I should really give Matt a hug. It really has been a pleasure, continues to be a pleasure to get to collaborate with Matt. You're lucky to have him there. I expected to see a little more Jayhawk blue in the audience. Congratulations. I will say I grew up in first Iowa City and then Northwest Iowa. University of Iowa lost in the first round, so I'm still feeling a little bit salty. I'll try to get over it. So I'm going to talk with you, have about 40 minutes to outline some, and in 40 minutes, it's difficult to cover all of what we can think about as we're imagining a future where we have some treatments in autism spectrum disorder that are based on biology, which is not obviously where we are right now. I'll also note that the other talks, which I won't necessarily get to see, look like they will fill in many of the gaps that I'll leave, and particularly pleased to see a really terrific set of speakers. I think it's always important to note potential disclosures or conflicts of interest. Some of what I'm going to talk about, because it's development of new medications, corresponds to work that has been funded by pharmaceutical companies. It's not possible to bring a new medication to market without a company involved. Where that is the case, you'll see that in red on the slide. Also want to note that this being an overview, I'm not going to spend time, not going to be able to spend time calling out all the individuals involved. And some of this work that I'll talk about today is from my group, and a lot of it is from others. Again, that'll show up on the bottom of the slide, and I'll periodically call out individual groups. So this is roughly the outline that I'm going to attempt to cover. And I'll note that I'm going to talk only very briefly about current treatments, in part because you'll hear more in the next talk. I think that the first thing to acknowledge, we have for decades now struggled with language, but even beyond struggling with the right terms to use to describe autism spectrum disorder or condition, how to refer to individuals, there's the simple reality that what we're describing is not a disease, it's not a unitary condition. It is multiple different risk factors that coincide in individuals that lead to different patterns that we lump together for simplicity and to give us ideas of how we might provide intervention. But that means that while somebody may present with enough, you can call them symptoms, you can call them traits, to receive an autism spectrum disorder diagnosis, oftentimes what that individual has to deal with in their life extends well beyond that. And so somebody may have a pattern that fits this, both medical conditions that are coming along with an autism diagnosis, sometimes biomarkers, sometimes actually more difficulty with intellectual disability or language difficulties than with the symptoms that led to an autism diagnosis, and sometimes a genetic condition that actually is explanatory, at least partially explanatory. And somebody else may fit a pattern like this. And you take this individual, and in one of the studies I'm going to tell you about toward the end, 288 other individuals, put them together and say that you're studying a treatment for a condition that we've defined. And I think we have to have that in mind as we evaluate potential treatments. When we just think about risk, most of our data from twin studies, from family studies, support a model where multiple genetic risk factors, in coincidence with some environmental risk factors, coalesce in an individual to contribute to risk. Most of the identifiable genes at this point are not of that sort, are not common, are instead quite rare, and contribute a larger portion of risk. So that includes Fragile X syndrome, which I'm going to talk about some as an example, but quite a number of other genetically defined syndromes. As we accumulate these, and we're now well over 100, we also can think of parallels in the environmental side, like exposure to certain drugs during pregnancy, like valproic acid, that have a similar pattern, which is to say they contribute enough risk that you can say with some confidence that if that risk wasn't present, an individual would not receive an autism diagnosis. Some of these coalesce to a degree around similar systems, but not most, or not in a way that leads easily to ideas of treatment that would extend to a broader group than, maybe a percent or two. When we think about the genetics, that's actually relatively simple, certainly in comparison with behavior. But genes, and now we have more than 100 that you can point to as contributing to risk, determine proteins which coalesce in networks, cells. By the time you get to synapses, you're actually talking about a scale of quadrillions. Those are, within particular brain regions, et cetera. And then at the top, obviously, behavior is infinite. We also have to take a developmental perspective. So genes can have different actions at different points in development. That is substantial complexity. But because we are using behavior to define what we describe as autism spectrum disorder, you can hope that as you track the impact of these genes up, and importantly, different impacts likely in different cells, different systems, but that should coalesce at some level underlying cognition or behavior, and that could potentially give us targets for potential treatments. Importantly, that could be a brain region, that could be a neurochemical, but that's really exciting to think about. We're going to talk about one potential example there, although it's not quite as sophisticated as all that. Most of the data, however, at this point coalesce at the level of protein systems and to a lesser extent cells, with substantial uncertainty between that and how you actually get to what we use in our diagnostic criteria. That's still a substantial improvement over our previous approach to developing treatments in this area, which is completely blind to what might actually be happening in the brain. And every medication that we use in autism spectrum disorder currently, and every behavioral treatment, is based on an approach that developed from coincidence or behavioral theory, rather than an understanding of what's happening in the brain. Adding to our complexity is the reality that in most cases where you identify a disruption of a gene that leads to autism risk, so decreased risk gene dosage, you then find later that increasing the dose of that same gene or genomic region also contributes to developmental risk. And sometimes you see autism on both sides of that. So if you had a treatment that made this individual better, you might actually make this individual worse on the other side of that inverted curve. So I'm going to talk very briefly about what we know about current treatments. You're going to hear more about this today, but I just want to use these as examples of how we come to knowledge about what works and how it works. On the behavioral side, and I'm talking about biology-based treatments, so I hope someday we can put those things together. We actually have quite a lot of data supporting early and intensive behavioral interventions in particular, and some data also supporting later interventions that are more targeted. The improvements that are seen are primarily for that early intervention class in cognition, so measured IQ, language, and adaptive function, not necessarily core symptoms of autism spectrum disorder. And importantly, it's been very difficult to show with any consistency that you can move those symptoms with behavioral treatments. Doesn't mean that they don't move, but difficult to show. And that's one of the challenges we think about testing biology-based treatments. So that's all I'm going to say on the behavioral side. I'm a little embarrassed by that. But importantly, I want to briefly overview, and you'll hear more next talk, the medication side. So it's instructive to reflect on both what we know and what we don't know. And I think it's particularly useful to think about where there have been a lot of studies. So prior to oxytocin, which I'm going to talk about toward the end, does anybody know? It's actually hard to do this in this room. What medication has had the most placebo-controlled trials in autism spectrum disorder? Raise your hand if you think it's risperidone or Risperdal. All right, nobody wants to raise their hand. It's hard to interact remotely in this sort of setting. It's actually secretin. And we now know with substantial confidence that secretin does nothing of any measurable benefit in autism spectrum disorder. But in the late 90s and early 2000s, this was a fad treatment that tens of thousands of kids with autism were exposed to. Based on anecdotal data, a small number of kids who'd received secretin as part of a medical procedure who seemed better afterward. And what we learned from this is, first, avoid fad treatments where possible, particularly when that involves an injection or other form of risk. But also in these studies, we learned that the placebo effect can be quite powerful in autism spectrum disorder, even though that seems quite counterintuitive. But this is an example, and this is a scale of total autism symptoms. This group measured lots and lots of different potential outcome measures. And what you see here is actually the placebo group shows really substantial improvement over time. So does the secretin group, but certainly you wouldn't take secretin over placebo in this particular scenario. Importantly, the majority of parents of kids participating in this particular trial were still interested in secretin as a potential treatment after being shown these data. And that's because our treatments have been so poor. Now there is more knowledge about the behavioral treatments that are available. I hope this would no longer be the case. But this is important to note, that it is oftentimes seductive to want to incorporate a treatment that has little data supporting it. And you can think about lots of examples of that today, and happy to discuss that in the question and answer session. Where we actually have knowledge is for associated symptoms. So irritability and agitation, you're going to hear about this in the next talk, but data for risperidone and aripiprazole, and then ADHD symptoms, again, you'll hear about this in the next talk. Substantial evidence that medications for ADHD also work for kids with ADHD and an autism diagnosis. Importantly, they work less well and in a smaller number of individuals. Where they work, they can lead to really substantial benefit, including benefit that extends beyond just ADHD symptoms, allowing kids to learn and interact with others more effectively. That's all I'm going to say about what we know currently. I took some out so that you could hear about it in the next talk. So I'm going to talk next about how we get from where we are right now in terms of knowledge to new treatments that are based on biology. I'm going to use an example of Fragile X syndrome. I will note that there are lots of other examples that we could talk about, and there is excitement for each of these that I'm listing here in terms of potential treatment development. Some of that I will also note is using a very different approach than what I'm going to describe today, which is using various different gene therapy approaches. I will say, at least for now, that's going to be reserved for really severe developmental disorders, including those that have risk of early death or substantial decline in function, like Rett syndrome. Fragile X syndrome in contrast, can have a really excellent quality of life for many individuals. It doesn't always lead to autism spectrum disorder. It does always lead to mild to moderate intellectual disability. The large minority of individuals will receive an autism diagnosis, but social difficulties are common even in the broader group. In younger kids, you see hyperactivity and impulsivity as well, usually meriting an ADHD diagnosis. And these kids oftentimes receive a genetic diagnosis, sometimes before they receive a diagnosis of autism, but sometimes are lurking in a population that's just described as having autism. And I will say I've made a Fragile X diagnosis in a 30-year-old, and people oftentimes say, well, I would be able to tell these kids based on how they look, but they actually look a lot like members of my family: prominent ears, prominent forehead, prominent jaw, and oftentimes blend. And so it's important to do genetic testing to evaluate whether somebody may have a Fragile X diagnosis. The genetics here has been worked out for a long time. I'm not going to spend time focusing on the details. There's an expanding repeat in the 5' and the early portion of the gene that, when it expands beyond a certain point, as it is passed down through families, leads to shutting off of that gene. And that impacts a number of systems, and I'm just going to show one of them by way of illustration. But the Fragile X protein is actually expressed downstream of a particular glutamate receptor. So if this is a synapse, this neuron is communicating to this neuron, the release of glutamate triggers immediate impact in the downstream neuron, but also serves to impact this mGluR5 receptor that actually serves as a brake. Tunes the synapse's response. And when you have Fragile X protein, that brake is present and prevents too much removal of this immediate response receptor, the AMPA receptor, from the cell surface. And that actually is very useful in tuning response. In the absence of the Fragile X protein, what you see is too much removal of this immediate response receptor, and then that synapse fails to mature properly. And so when that happens, you actually can see it under a microscope, that you don't suppress nearby dendritic spines, nearby attempts of the neuron to reach out and make a connection, because this synapse has failed to mature. And you can see that under a microscope in a mouse, and you can see that under a microscope in a human. And it looks essentially the same, which is really incredible to think about, that you can manipulate a gene in a mouse and recapitulate what you could see in the human disorder. Amazing. Unfortunately, the mouse's behavior is not obviously autistic-like. There are changes in hyperactivity. It's fairly subtle, changes in social function, but in tests that don't easily parallel what we do in a human. You put 2 mice in a tube and watch what they do. Impairments in learning that also do not offer a direct parallel. These mice do have inducible seizures, and seizures are seen in the minority of people with Fragile X syndrome. So it offers some opportunity to model. And importantly, we can do things in mice that we could never do in a human, like give a drug, in this case, shortly after weaning, extending all the way to sexual maturity. So this would be the equivalent of a decade. In a human population, and in a mouse, you can do that in 4 weeks. You can see that you rescue everything that you can point to as abnormal in that mouse. And this was work that was funded by Roche. I wasn't involved in it. But you can normalize, importantly, the appearance of these neurons under a microscope, normalize the hyperactivity, the learning, auditory sensitivity, even decrease the seizures, and no obvious ill effects on health. Really incredible to think about, exactly the sort of thing that we'd like to carry forward in a human population. That's really as far as we've been able to work this sort of cascade, to understand what's happening at a genetic level, work it all the way up to neuronal connections. But there is a gap when we think about the behavior. And that gap, importantly, makes it challenging to translate into a human. So unfortunately, the initial studies in humans done in adults and in adolescents did not show clear benefit for drugs that do the same thing, tune down response of this receptor in a human population compared to what was done in the mouse. And you can see in some cases, the gray line here, which is the placebo, actually outperformed the active drug. Really sort of discouraging. That isn't uncommon when there are associated side effects to a drug and no benefit associated to a drug. The placebo may actually work better. Why this happened? Well, I told you in a mouse, it was from weaning to sexual maturity. And here we're talking about adolescents and adults. So I think that's one of the big things. That isn't generally where we would start to intervene in a neurodevelopmental disorder. Doesn't mean we can't lead to benefit, but that benefit may be harder to measure, in particular because adults with Fragile X syndrome are oftentimes in quite varied settings that don't necessarily involve a lot of academic learning that parallels what would happen in a mouse model. And likewise, adolescents are oftentimes in life skills classrooms at that point. And I would say that both the age and the potential outcome measures, which here were disruptive behaviors that interfere with functioning, as well as social withdrawal, which isn't something that's easy to measure in this mouse model, may explain the lack of effect. There is now a child study that's funded. We're part of that study. Actually, the study is wrapping up. And that gives us the opportunity to drill down in a different way to think about development. This is in 3 to 6 year olds and looking at language learning during the peak of language learning in this population. And then drilling down some to understand potentially information processing with eye tracking, as well as circuitry with EEG, measuring the electrical discharges through the skull, but to try to understand how people respond to language in Fragile X syndrome. So I'm really hopeful about that. I don't have results to share with you yet. There are a number of other targets that we could think about in Fragile X syndrome that I'm not going to cover today. And I think importantly, this sort of approach, this approach that's usually described as precision medicine, I think most realistically may lead to profound benefit in Fragile X syndrome or in another syndrome. But it also offers an opportunity to learn how you study this sort of treatment. We've never had anything like this. So what might you detect? Is it language learning? Is it in 3 to 6 year olds? Is it something that needs to be even earlier? We don't know, and this is part of how we learn that. And then there's also the possibility that this will extend to a larger group that don't necessarily have Fragile X syndrome, but maybe have a similar difficulty that's happening at the level of neuronal communication. So I'm then going to talk about sort of the opposite. And this is where we've been to date. Our treatments in autism are really symptomatic, and that ranges from treating medical conditions, which are really important, to language interventions that show benefit for IQ, and then most of what I do as a child and adolescent psychiatrist, which is really treating co-occurring conditions in terms of behaviorally defined conditions in autism. But I would say also includes areas where some people with autism have difficulty and some people don't. Some autistic people are very sociable, very interested in others, but may struggle in social interaction. And some really have very little interest in others. So if we had a treatment that improved interest in others, that may help a subset, but certainly wouldn't help everyone. I would say this type of treatment is likely to benefit a bigger group than, say, in Fragile X syndrome, where that's likely less than 1% of people with autism. But that impact is less likely to be transformative. Certainly wouldn't be anything you'd describe as a cure. We're usually not thinking about that anyway, but probably wouldn't be something we'd describe as disease modifying, as changing the fundamental underlying difficulty, but may substantially improve an area of function. And it might fit a model sort of like this. And you can think in some ways that this is what we're doing with behavioral interventions. I'm going to use oxytocin as an example there. Oxytocin is clearly important for social function. It's known to be important for reproduction and sexual function across species. This is well known. You receive synthetic oxytocin sometimes to induce labor. There's also some data showing that it has a companion or sister or brother hormone in vasopressin that has some complementary effects. I'm not going to talk about that system today, but we could talk about it in the question and answer session if you're interested. The findings implicating oxytocin in autism are not strong. There's some inconsistent data around plasma oxytocin levels, some mixed genetic data. There are a couple of animal models of rare syndromes that have abnormalities in the oxytocin system. It's not clear that those are fundamental to the difficulties in those syndromes. But if you deliver intranasal oxytocin to anyone, you can change their social function. And it turns out that that's true in autism as well. So in this context, they defined folks as having high functioning autism spectrum disorder. Today, I would describe these as autistic people who do not have intellectual disability and have intact communication function. And so in this context, you see that one group received a placebo nasal spray, one group received an oxytocin nasal spray, and then there was a quote unquote healthy comparison group. Again, we would say neurotypical individuals today. This French group then used a variety of social tasks. This was a cyberball task where you throw a pretend ball to people you think are really on the computer on the other side, who either return the ball, throw it willy-nilly, or never throw it back to you. Most people will prefer throwing the ball to the individual who plays catch nicely. People with an autism diagnosis show no such preference unless they receive a squirt of intranasal oxytocin about an hour before this test. Now, that's really exciting to think about. This isn't how we make a diagnosis. This doesn't necessarily say that social function will change outside of the laboratory setting. A group at Yale then showed that you can also, and I'm going to skip past this quickly, you can also change brain response to social stimuli, to showing eyes to an individual in comparison to vehicles. And some of what you can see in the brain response includes areas of the brain that specifically respond to reward, suggesting maybe this changes social reward, although they did not see a change in behavior. And then there were quite a number of pilot studies. In this study, 35 individuals, half taking intranasal oxytocin for 4 weeks, half taking intranasal placebo. And there was an advantage measured on a scale of general autism symptoms for intranasal oxytocin versus placebo. But if you looked at it carefully with statistics, that was only significant when you accounted for baseline oxytocin levels in the individuals being treated. And interestingly, they saw that oxytocin levels in the blood actually went up whether you received oxytocin or received placebo, suggesting that oxytocin is actually involved in some way in the placebo response. So, as was true for secretin, tens of thousands of kids in this country then received intranasal oxytocin. And for the last decade, it's always been available for free shipping on Amazon. Now, this probably doesn't actually contain oxytocin, and the government, the Food and Drug Administration, will send a cease and desist letter to people who are inappropriately marketing potential drugs. So, about every 8 weeks or so, it's a different brand, but it's been consistently available for a long time. Clearly, what was needed was a large study. And the study I'm going to tell you about is a study of 290 participants across 7 major medical centers who received a sustained period of intranasal oxytocin treatment to try to avoid the period of initial placebo response at the beginning of treatment. These individuals were split into a group again, designed almost a decade ago, so just described as high functioning. Today, we would describe as having intact IQ and language function, and a group that did not have intact phrase speech, so that we were really covering the full spectrum of individuals, and from 3 years old to 17 years old. So pretty broad pediatric range. Importantly, the younger kids were not enrolled until we saw safety data in the older kids. Almost 6 months, so 24 weeks of treatment, and then an open label period of sustained treatment to see if response was sustained over time. Unfortunately, in data that were just recently published, we saw no advantage for the intranasal oxytocin versus placebo. So in some ways, this is a story similar to secretin, but importantly, you have to get to the point, this was a logical thing to pursue, but you have to get to the point where you robustly test a potential treatment. And you can see that most of the improvement in both the placebo group and the oxytocin group is in the first 4 weeks. And we're now looking closely to understand what predicts placebo treatment. But in this study, we did not have significant separation for the oxytocin. This doesn't mean that oxytocin should be discarded entirely, but it means that chronic administration of oxytocin should be discarded, should not be something that we're pursuing. If we think about oxytocin, we should think about giving oxytocin perhaps shortly before a behavioral intervention. I'm not suggesting that we do that. I'm suggesting that we could still test that. So now I'm going to pivot to how we might be able to do this better. And I would say this is conjecture because we don't yet have medications that are effective for core symptoms or core traits in autism. So one of those possibilities is to select individuals either who are more likely to respond or in whom we're more likely to be able to measure response. And so this is sort of asking, well, what if you had something that was more than Fragile X syndrome, but less than everyone with autism? And so I'm going to give you an example that targets the GABA system, the inhibitory system in the brain. I will say that there's some data pointing to the GABA system in autism and in particular mouse models with social deficits, including mouse models with genetic risk factors tied to autism. So there's reason to look at it. I'm not giving you this example because it's something that's effective. This is a study of arbaclofen. It's not available for prescription, but a study that had as its primary outcome in 150 individuals social withdrawal and did not show any significance for social withdrawal. The statistics are here for interest, but did have some signal for a clinician rating of global severity of symptoms. And so to just show the social withdrawal data, this is change from baseline. So you see everyone changes from their baseline at 4 weeks and at 12 weeks, both the placebo group and the arbaclofen group shows substantial improvement, but no difference whatsoever between the two groups. In contrast, if you look at changes for baseline in this clinician rating of global severity, you see that the arbaclofen group does outperform the placebo group. Again, this is not the primary outcome measure. This is not something you should go out and use. And it was right at the point where it's statistically significant only if you don't correct for everything that you've looked at or right at that threshold. But what you see is that there was a group of individuals who showed really a substantial change from baseline, a 2-point or 3-point or even 4-point change from baseline, which is quite unusual, but it's about 15% of the total. And so you see fewer people with no change from baseline in the arbaclofen group versus the placebo group. And that's what really jumps out at you. So this is not a general treatment, as I hope I've convinced you, for autism spectrum disorder, but we want to know who is in that group. That's 15% or so of the total. What predicts who responds? And so in this study, response was predicted by higher IQ or better language. But what we'd really like is a biomarker, some way to say, well, in this group that I can detect in a scanner or with EEG, they're much more likely to respond. That's a group that I want to test further. And so there is now a study that's going on, actually 2 studies, but with similar design, bridging from Canada to Europe, looking at response to arbaclofen and looking at EEG signal and looking specifically in the group where we saw initial signal that suggested response. So that's one way. And, we would like a biomarker that we can measure reliably, that can be well replicated. It could be genetic. It could be that it changes with treatment. It could be something that doesn't clearly relate. But what we really want is for something to fill this gap between precision medicine, big impact potentially in less than 1% of the total, and symptomatic treatments, small impact, maybe in a larger group of people. That's what we've got to date. I can't tell you what those biomarkers are going to be necessarily, but I would say that they may fit in a genetic category. They may be something like neuroimaging. I think it's more likely to be something like EEG because most kids with an autism diagnosis cannot lie still for a brain scan that takes 45 minutes, but they can have an EEG cap on their head. It could be something as substantial or easy to measure as body or head size. It could be an immune marker. It could be even something that you can measure as a risk factor in moms, like antibodies in moms who've given birth to a child with a later diagnosis of autism. But I think that that's one way that we're going to improve how we're targeting our treatments and have a more feasible pathway to treatment development. I'd say another way is around outcome measures. So I've told you briefly about some of the outcome measures in these trials. Oftentimes it's simply parent report, sort of checklist. These are things that my child is dealing with that are difficult. Sometimes it's a parent interview, which is a little more robust, where the parent and clinician together can say, well, this is an area of difficulty, and this is where we would put it in how difficult it really is in comparison to other kids. Sometimes it's a clinician rating. Interestingly, some data suggests that parent rating is better. Some data suggests that clinician rating is better. We don't know at this point, but in most studies, you do both. And sometimes you use an exam, a formal evaluation of the child, like the Autism Diagnostic Observation Schedule. And there are some adapted versions of an interaction with a child that offer fine-grained ratings of social function that may be more appropriate over time. These sorts of approaches have led to an overall placebo response in autism that in at least larger studies tends to hover around 25%, sometimes as high as 50%, which is a real challenge. What we'd really love is to have objective outcome measures that are not susceptible to placebo response. And that could include something like eye tracking, where you're able to watch somebody's social response in real time. Different results are seen in autism depending on the age of the individual and where it's being done, other characteristics of the individual. It could be EEG. This could be a biomarker for identifying who could respond, but it also might show response. And there's some hope that looking at, for example, response to faces using EEG could give us some opportunity to see at least differences between individuals and maybe response to treatment. We could think about neuroimaging, and I would say that that potentially is a quite robust measure, but only the minority of individuals can participate. And that's really challenging when you think about a treatment study versus a study that's trying to understand how brains may differ in autistic people versus neurotypical people. Oops. I would say one other opportunity would be to think about learning as an outcome measure. And I mentioned in that Fragile X study, we're looking at language learning. But this is what's moved in early intervention studies in autism. IQ is usually the most robust difference. And this is an example using the Early Start Denver model. You can see that IQ robustly separates between a group receiving the active intervention versus community-based control. So ultimately, as I mentioned at the beginning, you'd like to come back to use what we currently know at a behavioral level. And this is really the only example of where that's been done in autism to date, where there were two groups enrolled. Both groups had substantial difficulty with parent ratings of disruptive behavior in the home. So you can think about these as kids who have difficulty with tantrums, kids who have difficulty sometimes with aggression, self-injury, disruptive sorts of behavior. Both groups of kids randomly assigned, both groups received risperidone. And you can see both groups have had substantial improvements. And one group received a parent-mediated behavioral intervention. That's the group in blue. You see that that group is doing better in terms of parent ratings. The other group received a psychoeducation, sort of a teaching intervention, and improved less. Unfortunately, when they went back and looked at these kids 6 months later, there was no longer any significant separation between the groups. But also importantly, the group receiving the parent-mediated behavioral intervention ended up on a lower dose of risperidone, which, looking back, you'd like to change that and have everyone have a more common experience of medication and see what additional benefit behavioral intervention offers on top of stable medicine. But that is another indication that behavioral intervention works, but illustrates also some of the challenges in these studies. You'd prefer to see instead something like the story with D-cycloserine, which people may or may not have heard about. In anxiety disorders, D-cycloserine has been shown now across multiple studies, from mice to rats to humans, to potentiate response to psychotherapy. That if you give this drug, which acts on that same glutamate system, roughly an hour before therapy, you see that people are able to learn that they can handle something that makes them anxious much more quickly. And you see that that response rate is different here than here, even though there is no longer such a big change at the end of treatment. This is obviously a very small study. So that's where we'd like to get to, putting the brain in a state better able to benefit from our behavioral treatments. I'm not going to read the summary slide. I will say sometimes hearing these stories feels depressing because I'm not telling you about anything that works right now. My brother is an oncologist, MD, PhD oncologist, and my mom received the benefit of oncology interventions that were based on a biological understanding of her particular cancer, lived probably 14 years longer than she would have otherwise. And so I'm always reminded of how knowing the biology informs treatments, usually with a gap of about 20 years in cancer, which is a little depressing. That gap is narrowing. And I think we can hope that the revolution in understanding of the genetics of autism that's happened just in the last 5 years or so will yield treatments. I can't tell you exactly when those are going to start, but I think within the next 5 years, we will have some of these precision medicine treatments. And we may have other treatments that are based on an understanding of biology in a smaller group of individuals than the whole, but maybe a larger group than just less than 1% at a time. So there are quite a lot of folks involved in these different studies. I'm not going to call them all out individually, but we'll also acknowledge folks who've provided funding over time. And I should stop there and give time for questions. Thank you. So I will say I grew up in Iowa. And when I went to medical school, I was told I spoke really slowly. Was sometimes criticized for it. And I've lived in New York now for 7 years, and am now told that I speak too quickly. So my apologies if some of that goes quickly. It's meant to be conceptual and not meant to be catching all the detail. Thank you.
Thank you, Jeremy. I guess it's all relative. Yeah, you can see me now. Okay. I didn't realize that in medical school they teach you to speak faster too. I mean, it's I guess that's
doctors don't like to listen, and that's true when it's a medical student.
To be clear, MD. I think you're talking about MDs, not PhDs. So
yes.
We can have a discussion later. All right, so we're going to open it up for question. And Jeremy, in this format, I will just relay the questions to you, so you're able to get the gist or hear all of the questions from the audience. I did just want to open it up because, first of all, you covered a tremendous amount there, and it was both, I think, encouraging and overwhelming in terms of the challenges that lie ahead in terms of developing biological therapeutics and new, more precise and effective ways of supporting autistic individuals. I put the emphasis on encouraging versus overwhelming, but it was wonderful. It also presents all of the many factors that really make this such a difficult and challenging endeavor. And so thank you for being a leader in this area, but also it is a reminder of just, how many different details you have to sort through to develop more effective therapeutics. One point just to start us off, and then I'll open up to further questions, is, you described some of the behavioral intervention strategies, and I know there are a number of behavioral interventionists, behavioral providers within the audience, and you referenced this a little bit at the end in one of the studies, but I wanted just to hear your thoughts on how some of what we do know and what we can do in terms from a behavioral perspective may be combined or leveraged in some of these clinical trials. And I think about this in the context of even like the oxytocin trials, just thinking about, a therapeutic that's intended to enhance social behavior, to change social behavior. Social behavior is so complex, right? And so it's, even if someone becomes more, develop a stronger inclination to interact socially, developing those skills requires additional supports. Or behavioral strategies. So I wondered how that might be leveraged, might be addressed in terms of both clinical trial study design and just your overall thoughts on that regard.
Yeah, no, I think that's a really important point. And as I noted for oxytocin, and I don't mean to say that that's specific to oxytocin, but that if it is capable in a laboratory setting of changing, somebody's desire to play catch with somebody, for example, maybe for a subset of kids who are not that interested in engaging, say, in social skills treatment, that could be transformative, right? And so you put those two things together, it could be quite helpful. For other kids, it may not offer much benefit, right? The issue isn't a lack of interest, it's sort of figuring out how to negotiate that. And it may be actually making them more interested in others makes it more difficult to implement the social skills training that may be helpful for that particular child. I will say the area I see this more frequently in the clinic currently is ADHD and autism. It's quite common in autism. And I will say ADHD is not that hard to treat. And there's about a 50% response rate for each of not that hard to treat from a pharmacologic perspective. So it doesn't take a rocket scientist, that is to say. There's about a 50% response rate to each of our major pharmacologic classes. Very frequently, kids don't get considered for treatment of their ADHD until they've already failed multiple behavioral interventions. And then often they've lost access to some of the interventions that may provide benefit once their ADHD is adequately addressed. And that's a real problem. And we have to figure out how to partner together more effectively, how to think in a multidisciplinary way about at what point do we say that a child isn't responding to a treatment because of their ADHD, and then provide intervention so that we don't have a kid who's going through 2 years of what should be effective treatment, but never able to focus on it. And then they finally get their ADHD treated when they're 8 years old. But at this point, they've lost opportunities to communicate and integrate in a classroom that they otherwise would have had. So I think that is meaningful, and nobody has adequately addressed it.
Yeah. I'm going to open up to the audience. So if folks could just raise their hand and let us know the question, then I'll relay that to Dr. Veenstra-VanderWeele. Question over here. So Jeremy, I'm not sure how much you heard of that question.
Unfortunately, none of it. I'm
sorry.
Oh,
okay. So that's quite a case. There's a very eloquent question from Christine, who has 2 autistic sons, and is, I guess, to summarize, I think where the gist of the question is the extent to which we as a field, that there are studies existing that have really looked at all of those different environmental contributions to some of the challenges faced by autistic individuals, as opposed to just treating or changing the autistic person, but what do we know about how we can develop the skills and the supports amongst the educators, the different providers, caregivers that also may contribute to really a better overall quality of life and outcome for autistic individuals. Is that right, Christine? All right, I did okay.
Yeah, okay. That is a great question and or point. But I think not nearly enough. And part of that fits into what people sometimes describe as the double empathy problem. So some people will say that autistic individuals struggle to understand others, and you could say struggle to understand non-autistic individuals. And in reverse, I think that we don't know enough, haven't worked clearly enough to help folks understand what an autistic individual's thinking, experiencing in turn. And I do think that adapting the environment to fit the individual is what we do in lots of areas, and is some of what we do reliably in classrooms, in adult learning environments, but we haven't studied that terribly well. And I would say it's an area of disconnect that is absolutely worth further study.
Yeah, I think that's well said, Jeremy. I guess though I was looking around for Linda Heitzman-Powell, who I don't see, but I'll highlight some of her work later on. But her OASIS program here as well is really aimed at developing some of these, some of the evidence-based skills amongst educators and different providers. But it's a great question, Christine. I think I agree very much, Jeremy, with your sentiment that it's an area that we could really amplify or enhance moving forward in the research domain. Jeremy, do you have time for one more question?
You bet. Yep.
All right. So any other questions in the audience? Yes.
I'm wondering if the biomarker area of study is addressing gender differences.
Yep. So the question is around biomarker development and the extent to which sex differences are integrated into those analyses and studies, and maybe even what we know about that. Yeah.
That is I would say again, a great question and also point. I think I suspect that in asking the question, you kind of know the answer. Not adequately. So there have been some studies recently, and not all of that work is published, but that have over-enrolled autistic women or girls with an autism diagnosis in order to really have a clearer sense of that. Most autism research is heavily biased toward males in the same way that autism diagnosis is heavily biased toward males. And some of that, we think most of that, I would say, is driven by biology. But some of it is driven by our shaping our diagnostic criteria and our diagnostic instruments by most of what we've seen, which is in boys, and missing diagnoses in girls. So there isn't strong evidence that any particular biomarker is, say, present more in boys than in girls, but there are hints. So elevated blood serotonin levels, which have been sort of known as the longest standing biomarker in autism, are seen more often in boys than in girls. We don't know why. There's also data that many of the biomarkers, and none of it particularly strong data, but many of the biomarkers that are associated with IQ are also associated with sex, or I guess easiest way to describe it is sex instead of gender in autism, such that IQ on average tends to be lower in girls with a diagnosis of autism than in boys. So any biomarker that's associated with IQ is going to show a sex-based skew, if that makes sense.
And I would just add too that there has been, in an encouraging way, there's been an increased focus and attention to important sex differences within autism amongst biomarker research programs. There's been a large Autism Center of Excellence that has been funded for the past 5 years, I think, maybe 10.
Yeah, I think it's 10 years now. Yep.
Yeah, 2 cycles. Specifically focused on imaging of girls with autism. So more information will be known about that, I think, in the near future, but it's certainly, the field is still a little bit behind in that regard. With that being said, Jeremy, I wanted to thank you again for joining us, for providing a wonderful overview, and frankly, it was just great to see you. So thank you for serving as a keynote for this meeting. Real quick, just our upcoming talks are just a couple minutes away, and they'll be down the hall. We'll also have a poster session in here starting at 9:30. Poster presenters will be in this room with their posters put up, so please join. And otherwise, I wish everyone a wonderful day, and thank you again for getting us started, Dr. Veenstra-VanderWeele.
Thanks so much, Dr. Mosconi.